right, folks, I think it's time we get started. Once again, there are some openings in the freshman bio labs for folks just like you to help and set up the labs so that folks just like you can take the labs starting in the spring semester. So if you're interested, that's the story. You can mention your interest to people in the lab when you're there this week or next week if you like, and they'll be able to tell you how to apply and all that stuff. All right, so we got that one announcement. There was another announcement that I mentioned on Tuesday, and that is that there was a free lunch in the Student Union Atrium yesterday. I was there. Where were all of you? Right? I didn't see anybody else that I recognized from this class, at least, over there. So I got my free hot dog and I left because I didn't want to be lonely. And I have one other sort of announcement for you today. At 10:15 this morning, Wright State is going to be conducting a first ever earthquake awareness kind of experience. And frankly, I don't know what to do about that. You know, it's smack dab in the middle of this class. And certainly, we want to be prepared in the event of an emergency, but I'm wrestling and going back and forth in my head even as I'm talking with you right now about what it is we should do when the alarms start to go off at 10:15. So one option might be to scream and run around in circles. I don't think that's part of what it is they had in mind. I can tell you these things. We are in a part of the world where earthquakes can happen. The thing is, it turns out that we're in the middle of a plate, such a tectonic plate, such that if we had an earthquake, it would be a really big one, and it would probably be very difficult to survive. So there's one thing to keep in mind. And in the event that there is an earthquake, you probably want to take actions pretty quickly to have some small chance of surviving. The sorts of things that you might want to do are go to the walls, all right? So the walls are likely to be a better, safer place for you to be than in the middle where you are. There are tornado shelters in the hallways that would be very helpful. So the rules are, well, the words of advice for earthquake preparedness is that you want to get low. Before the earthquake knocks you down, you want to get yourself close to the ground. And if you can get under something, you know, I have this convenient earthquake shelter right up here, this podium. I don't know what's going to happen to the rest of you guys. Yeah, there'll be a mad dash for the podium. And there are those external walls at the edge. So let's just play it by ear. We'll see how the lecture is going at 10:15 and decide what we'll do at that point. But just be ready. At 10:15, there will be a test of our earthquake preparedness system. All right. So, at least until the earthquake hits, let's talk a little bit about some biology. And where we're at is I'm kind of halfway through the process by which cells extract energy from things like sugars. I feel like I'm shouting today. Is the microphone louder than usual? Is it just me, hyper aware of my own voice? So we're halfway through talking about catabolism. On Tuesday, I'm going to talk with you about the flip side, the kind of the other half of metabolism, and I'll talk with you about anabolism. But for today, we're going to finish up talking about catabolism. You'll recall that on Tuesday, I talked with you about glycolysis, so the 10-enzyme process by which organisms, prokaryote, eukaryote, archaebacteria, by which all organisms convert glucose, a 6-carbon sugar, to pyruvate, a 3-carbon sugar, and along the way generate 2 NADHs and also have a net gain of 2 ATPs. So there's a 2 ATP investment, a 4 ATP recoupment, net gain of 2 ATPs. For some organisms, for those that live anaerobically, that's it. That's all the energy you get out of that 686 kcal per mole glucose molecule. But for those who can do aerobic respiration, there's more that can happen. And that's part of what we'll be talking about today. So this is the same lecture outline that you had seen on Tuesday. The slides are a little different because we're halfway through. But as a refresher, let me point out a few of the highlights from Tuesday's lecture. I didn't show you this picture on Tuesday, but I alluded to this concept. I tried to make the point that glycolysis is not just a really ancient pathway, something that living things have worked out very early in the evolution of life and have been working with pretty much unchanged for a very long time. But not only is it very ancient, it's also very central to the rest of metabolism. So the rest of metabolism sort of gets tacked on to this existing very important pathway that we call glycolysis. And this slide makes that point, right? What do you do with a carbohydrate? Any kind of sugar, what you do is you make carbohydrates, you break them down into sugars, and then you channel them through glycolysis. So if you eat a sugar that's something other than glucose, you know what you do? The first thing you do to get energy out of it is you convert that sugar into glucose. So there's all this stuff in metabolism whose business is to convert sugars into glucose so they can run through glycolysis. What about a fat, right? Fats are good for energy storage, energy transfer sorts of things. If you got a fat, you know what you do with a fat? You in some way slip it in, get it channeled so it gets into either the middle of glycolysis, entering here with glyceraldehyde 3-phosphate, or bring it in as acetyl-CoA so you can run it through the citric acid cycle, which we'll be talking about today. Same thing for proteins. If you wanted to get energy out of a protein, what you do is you can convert parts of those proteins into pyruvate. You can convert some proteins into acetyl-CoA, and some can go directly into the citric acid cycle. So anything that any living thing wants to get energy out of gets channeled in some way through this catabolic process, initially with glycolysis, and then later on with the citric acid cycle. And these reactions that we're talking about here can usually go in both directions, right? And so if you needed to make a protein, it's usually possible to shunt things out of glycolysis and run this the other direction so that you can take pyruvate and use it to make amino acids, or you can take acetyl-CoA out of this pathway and use it to make amino acids. Or you can take acetyl-CoA out of the pathway and invest it into making fatty acids and lipids. So once again, this is a very old pathway, but it's very central to all of metabolism. All right. Then, in a nutshell, this is where all of this stuff plays out within a eukaryotic cell. Remember, as eukaryotes, we have these internal membrane-bound compartments, and those internal membrane-bound compartments are specialists. They're dedicated structures and chemical environments to being able to allow certain chemical reactions to take place in them better than they might have taken place elsewhere. In the cytoplasm is where glycolysis takes place. So all 10 of those enzymes that we were talking about in lecture on Tuesday take place exclusively in the cytoplasm. Those enzymes are never found in any other compartment inside of a cell. But the things that we're going to be talking about for the most part today, the citric acid cycle and oxidative phosphorylation with the electron transport chain, those things happen exclusively within the mitochondria. And along the way, take note, I'll try to draw your attention to it, but take note of how it is that the structures of the mitochondria play an important role in facilitating those reactions as they take place. So once again, in biology, structure and function are very intimately related. You're going to see some molecular level examples of that as we talk during the course of today's lecture. The bottom line is this. Right? From 50,000 feet, looking at what we're talking about here, the whole process of extracting energy from glucose is in some sense a slow burn. What we do is we take that 6-carbon glucose molecule, combine it with 6 oxygen gas molecules, and end up at the end of the whole process, when all the energy that could be extracted from the glucose has been extracted, at the end of that whole process, you get 6 carbon dioxide gas molecules and 6 water molecules, as well as the release of quite a large amount of energy. So that's what this whole landscape looks like at 50,000 feet. We got halfway through the process during the course of Tuesday's lecture. We're going to go the rest of the way today. Zooming in on it a little bit more, now it's 20,000 feet, what you can see is this happens in steps. The first step has a name. We call that first step glycolysis. Again, 10 enzymes to catalyze this series of reactions that we call glycolysis. And the bottom line for glycolysis is this. You take glucose and you convert it to pyruvate. And along the way, you capture some ATPs and you also generate some NADHs. And again, this will take on more significance as we go through today's lecture. 10,000 feet, right? Now we're zooming in a little bit more at the details of what goes on with glycolysis. As a general strategy, what glycolysis does is this, is it primes the pump. There's an energy investment phase to glycolysis where ATPs are actually consumed to get the glucose molecule into a more reactive state so that it's in a better position to run through the rest of glycolysis. And after that energy investment phase, you're off to the races and you're into the energy payoff phase where ATPs are formed and NADHs are formed, as well as the pyruvate and the water. Now, I'm not going to take the time that I did on Tuesday to go through each of these different reactions. I'm going to flip through them fairly quickly right now. But things that you should be keying in on as best as you can. Look at the molecules and get a sense as to whether or not a molecule has lots of energy, a medium amount of energy, or relatively little energy, right? And so on these slides, as you go from left to right across the screen, generally speaking, you're going to be having molecules that have less energy, but there are certainly some interesting exceptions to that rule. Glucose is certainly an energy-rich molecule, but when you tack that phosphate group onto it, that makes it even more energy-rich, right? So keep track of the energy. I'm hoping that at a glance you could look at one of these structures and say, gee, this probably has more high-energy covalent bonds associated with it than this does. I also hope that you won't be intimidated by the names of the enzymes, right? I told you about 3 different classes of enzymes that you'll see popping up time and again, not just in glycolysis, but in a little bit in the citric acid cycle. There are the kinases, there are the isomerases, and there are the dehydrogenases. Kinases have the job of moving phosphate groups from something to something else. Often ATP or ADP is one of the somethings. Isomerases or mutases, equivalent terms, isomerases or mutases simply make chemical isomers. Right? So you have a compound that has a particular chemical formula can be changed into another compound with the same chemical formula, just a different deployment of the covalent bonds. Right? So it's an isomer, a structural isomer. And dehydrogenases have the job of moving hydrogens from something to something else, typically involving either FAD or NAD. And again, we'll talk more about that as we go through today's lecture. So here you go. You got the first 2 enzymes. Their names make some sense, especially if you know about those 3 classes of enzymes. I don't think that the names of these compounds should scare you too much either, right? The 6-phosphate at first might sound like Greek, but it's pretty straightforward. It's literally the 6th carbon in the chain has a phosphate group attached to it. And there's no easy way to remember this aside from saying, gee, that's glucose, right? That's blue is blue, glucose is glucose. It's one of those sorts of things. It's a tautology. Moving down the process of glycolysis. Remember, we got 10 enzymes that are going to be involved here. We've got another kinase that kicks in, meaning that there's a phosphate group that gets transferred. We've got an aldolase that breaks this 6-carbon molecule up into two carbon molecules. There's an isomerase that converts one of those 3 carbon molecules into the other. Notice that the arrow goes both ways, but here's the thing. These get converted into something else, so there's almost always a general flow from here to here because these guys are getting changed into other things. So there's less opportunity for this to move back to that. But the isomerase can make it go in either direction. And then you get NADH involved or created here. No big surprise, right? That means there's a dehydrogenase that's involved. Some more phosphates get tacked on as part of that process. Some ATPs get generated by a kinase as we're extracting energy now as opposed to investing energy. There's another isomerase or a mutase that moves things around a little bit. And ultimately, we have one last kinase whose job it is to create one more ATP. Fancy term for that kind of ATP creation. Remember, that's called substrate level phosphorylation. So at the level of the substrates, you're making ATPs by phosphorylating ADP. Okay, so that's substrate level phosphorylation. In a little while, we'll be talking about a different way to do phosphorylation. It's going to be oxidative phosphorylation. It's a different way to do it than what we see here with substrate level phosphorylation. So there you go. There's the Reader's Digest version of Tuesday's lecture. At 10 or 20,000 feet, glycolysis has an energy investment phase where 2 ATPs get consumed, then an energy payoff phase where a net gain of 2 ATPs are realized, and also some of these NADHs. All right, so there we go. We're up to this point in the lecture. Brace yourself, it's time for the citric acid cycle. When I was a kid, we didn't call the citric acid cycle the citric acid cycle. We called it the Krebs cycle. So you'll still hear it called that sometimes. The guy who discovered the Krebs cycle, what do you think his last name was? Krebs. So that's where we get the Krebs cycle from. Krebs was a pretty modest guy. He knew of the embarrassment that was visited upon Davison and Danieli. Remember the guys who were a little bit off the mark with phospholipid bilayer and membrane structures. And so from the get-go, he insisted that the Krebs cycle be called the citric acid cycle, and it's partly to honor him that people today call it the citric acid cycle, even though he's the guy who worked out all the details. He spent his career figuring out how it is you convert pyruvate into carbon dioxide gas, right? How it is that this cycle works. And he ended up discovering that there were a series of 8 enzymes that were involved. So in the same spirit as what's going on with glycolysis, a 10-step process, this last part of extracting energy from glucose, going from pyruvate to carbon dioxide gas, this is an 8-step process. But instead of being a linear process, it's actually a cycle. There's a little churn of a wheel that's going to be happening. You'll see the pictures in just a moment. So again, bottom line for what the citric acid cycle is doing is we're converting the end product of glycolysis into carbon dioxide gas. And you can quibble as to where it is the citric acid cycle begins. It could either be this process of starting with pyruvate, or you could do the next step. There's a 1-step going from pyruvate to acetyl-CoA. So some people will tell you the citric acid cycle is acetyl-CoA to carbon dioxide. Others will tell you it's pyruvate to carbon dioxide. It's just words. It doesn't have anything to do with what's actually happening at a cellular level. So let's see what's actually going on here. Here's pictures for the words that I've just said. So again, what's going on is we want to get our pyruvate, that what gets handed off at the end of glycolysis, we want to get that pyruvate converted into carbon dioxide gas. And the name of the game really is, while we're extracting and collecting in a useful way as much of the energy as is possible. Okay? So here is that 50,000-foot view of what we'll be talking about. We start with 2-pyruvate. Why 2? Well, it's 2 because pyruvate's that 3-carbon molecule. Remember, glucose is a 6-carbon molecule. For every glucose molecule, you will make 2 3-carbon pyruvates. Right? So one of the tricks to keeping up with what's going on here is counting the carbons. Remember, we had 2 pyruvates for every glucose. Those pyruvates are 3-carbon molecules. They get converted into 2 2-carbon molecules and 2 1-carbon molecules. The 1-carbon molecules are carbon dioxide gas, right? But where the energy is going to be is in these acetates. In the acetate, the 2-carbon energy-rich molecule gets attached to something called coenzyme A. Pretty big, bulky thing that just gets reused over and over and over again. Okay, so acetate gets attached to coenzyme A to make acetyl-CoA, and then this 2-carbon molecule gets hooked up with a 4-carbon molecule, oxaloacetate, to make a 6-carbon molecule called citric acid. And then you're off to the races. You go through the whole cycle, you convert that 6-carbon citric acid back into the 4-carbon oxaloacetate, and you're ready to hook it up with acetyl-CoA and do the whole process over again. As you go through the cycle of the citric acid cycle, some ATPs get made. And as you go through the cycle of the citric acid cycle, some NADH and some FADH2 get made, as well as some carbon dioxide gas. Okay? Where the energy is getting pulled off in a way that cells will be able to use is certainly in the form of the ATPs, but then also there's energy in these high energy electrons associated with NADH and FADH2 that can be extracted through the electron transport chain, which is what I'll talk with you about at the end of today's lecture. All right, so that's the citric acid cycle from 50,000 feet. Let's get ready to zoom in and see a little bit more about what's happening at the grassroots level. Why is that slide there? What's up with that? We're done with glycolysis. Here's why this slide is here. This slide is here is because I just want to draw your attention to the fact that at the end of glycolysis, right, this is the molecule that gets created, pyruvate, a 3-carbon molecule, right? Glycolysis is done. This is all cytoplasm stuff. So we're hanging out in the cytoplasm with pyruvate. We're making more and more pyruvate. The more glucose that gets metabolized, the more pyruvate is going to be piling up in the cytoplasm. That pyruvate needs to be shunted into the mitochondria in order for the citric acid cycle and the electron transport chain to come into play. Right? So the pyruvate has to be physically transported across that mitochondrial double phospholipid bilayer that the mitochondria have. And there are proteins embedded in the membrane of mitochondria whose task it is specifically to give a one-way pass to pyruvate. Lets pyruvate go from high concentration in the cytoplasm to low concentration in the mitochondria. Right? Passive diffusion is going to get this pyruvate from the cytoplasm into the mitochondria. As the pyruvate passes through those pores, carbon dioxide gas is removed. You can see, so this is pyruvate. They're drawing your attention to the the red ones that stick around, and the gas that bubbles away. Right? So here's a CO2 that's going to get clipped off, and you're going to be left with this high energy molecule here. And coenzyme A gets linked to the acetate to give you acetyl-CoA. Notice there's another enzyme that's involved here. It's a three-step process, right? You transport the pyruvate into the mitochondria. Waiting to receive it is an enzyme that is a dehydrogenase that converts NAD to NADH. And then also another enzyme comes along to attach coenzyme A to acetyl-CoA, and you get that structure. Coenzyme A, it's just a space keeper, right? It gets used over and over again. Don't pay any attention to that, right? The balls that you want to be keeping your eyes on here, where the energy's at, is in that acetate molecule. So that's how we get pyruvate into the mitochondria, and that pyruvate ultimately gets converted in the process to acetate, and the acetate gets some energy taken off of it and gets attached to coenzyme A. So here is that previous slide, again shown in pretty much the same level of detail as on the previous slide. And what's going to happen is we're going to take that acetyl-CoA, we're going to kick off the coenzyme A, and the acetate is going to get hooked up to a 4-carbon sugar to become a 6-carbon sugar, and we're going to go through this process. This particular slide is showing you where all of the carbon dioxide is getting released. You'll see that the CO2s get kicked off pretty early in the process. You'll see that after the CO2s get removed, there's a dehydrogenase that's going to be generating some of these energy-rich NADHs. Then there is a substrate-level phosphorylation that takes place over here. And one more dehydrogenase does its thing at the end of the process before you're back to starting it over again. Well, guess what? Now we get to talk about each of these different steps. Here they are all together. And, you know, I'm not sure what to tell you at this point, aside from the fact that there's a lot of chemical structures here, right? I'm not going to be expecting you guys to memorize the names of these different chemical structures. Your textbook, I think, does you a disservice by not giving you the names of the enzymes. They give you the names of the chemicals, but not the names of the enzymes. In pictures like this, I think you guys can know what most of these enzyme names will be, right? There is a dehydrogenase involved here. There's a kinase involved here. There's a dehydrogenase, another dehydrogenase, right? So most of the guts of the names are things that shouldn't be too intimidating to you. I said a moment ago that I'm not going to expect you guys to memorize the structures and associate them with their names, but I think it comes up often enough, and it's a big enough deal, that it's probably good for you to know that the 6-carbon molecule at the beginning of the citric acid cycle is citric acid. All right? Or if you prefer, the salt of citric acid, which is called citrate. Any acid's salt, you clip off the acid and you tack on 8. That means it's the salt of an acid. So citrate is the salt of citric acid. All right? So I hope you could, I think it's reasonable. I hope you don't think I'm being unreasonable when I tell you it would be good if you knew that the beginning of this process starts with citric acid, right? And what makes citric acid sort of special is that it is a 6-carbon molecule. You can see there are these 4 carbons that come from oxaloacetate. There are 2 carbons that are coming from the glucose that got converted to the pyruvate, that got converted to acetate, to get tacked on to citrate. All right? So the first step in the process involves a 6-carbon molecule called citrate. I hope you'll remember acetyl-CoA acetate because it's got those 2 carbons that came from the glucose. So I hope you'll remember acetyl-CoA, that name. And I think it would also be fairly reasonable for you to recognize oxaloacetate because of its important role in this whole process. Oxaloacetate is the 4-carbon molecule that the acetate gets tacked onto. 4 plus 2 equals 6. The 6 is the 6-carbon molecule called citrate, or citric acid. So those are the only 3 that I think is particularly important that you guys get under your belt at this point. The rest of the names aren't that scary. You know, isocitrate is just an isomer of citric acid, right? This one kind of rolls off the tongue. I'll pronounce it for you so that when you use it in social settings, you won't, mispronounce it. That's alpha. The little squiggle at the beginning is the Greek letter alpha. That's alpha-ketoglutarate. It's got a nice roll to it, doesn't it? You could write poetry using words like alpha-ketoglutarate, right? Next in line is succinyl-CoA, right? Then succinate, and then fumarate, and then malate, and then oxaloacetate. Back to the process at the beginning again. So that's how you pronounce each of those names. I don't want you to, never have heard the way that they're pronounced, but I don't think you need to commit to memory that alpha-ketoglutarate is the next step in the process after isocitrate, and I don't think you need to commit to memory that this is the structure of alpha-ketoglutarate. So that's stuff you don't necessarily need to know for this class, for the test coming up in November. Let's talk about the stuff that maybe you should know. Things you should know: acetate, citrate, oxaloacetate. Check out what's going on here. And the colors end up being pretty important and good to look at to help you keep track of what's happening. The ones that are shown here in blue in oxaloacetate, those are the ones that ultimately are going to get lost as carbon dioxide gas. Okay? The ones that are shown in red are the ones that are coming from the glucose. So these are all things that Hans Krebs had to figure out as he was working out the citric acid cycle. How do you suppose he figured that stuff out? In a word, radioisotopes. He had radioactively labeled carbon, that he was carbon 13, carbon 14, and he was able to track what was coming off when, in terms of when does this show up, when does a radioactive label show up in CO2? Is it right away when you feed the glucose, or does it take a little while? And so check out what happens. You take your 2-carbon molecule, you hook it up to this 4-carbon molecule. You specifically hook it up right there onto that carbon. So you got the 2 acetate carbons up here, and the oxaloacetate is pretty much unchanged down there. Water comes in, water comes out, right? You do some rearrangements. Guess what? This enzyme has isomerase as part of its name. And you end up to make isocitrate that's pretty much the same as citrate, just a little bit of a rearrangement, right? The hydroxyl is down here now as opposed to up here in the middle, and that's about it. Right? So subtle change in the structure to go from citrate to isocitrate. A dehydrogenase takes some high energy electrons off of the isocitrate and releases a low energy carbon dioxide gas. You had 6 carbons up here with isocitrate. Alpha-ketoglutarate has 5 carbons because one of those guys went away as CO2. Okay? Check out this structure compared to this structure. Notice it's this one that gets clipped off. You're effectively getting the energy out of this bond to make that NADH. So you lose one of the CO2s, and you've got an energy-rich 5-carbon molecule. If I were to show you this structure and ask you, does it have more or less energy intrinsic to its covalent bonds than this structure, I hope you guys would pretty much, without missing a beat, say, gee, This has more energy associated with it than this does. This has more carbon to carbon, carbon to hydrogen bonds than that does. 6 carbon versus 5 carbon. It's going to be tough for a 5 carbon molecule to have more energy than a 6 carbon molecule, but even if you didn't do that kind of bookkeeping, there's more high energy bonds with the isocitrate than the alpha ketoglutarate. Then check out what happens next. Another enzyme, a 4th enzyme, comes in. That enzyme's a dehydrogenase. It's going to release another one of those CO2s. So now we're back to a 4 carbon molecule again already. This dehydrogenase, though, is going to, as it releases the CO2, take off some high energy electrons and make some NADH for us. And now you're back to a 4 carbon molecule. Notice that the 2 of the 4 carbons in the oxaloacetate have been released as CO2. It's not the pyruvates, red carbons, that get released as CO2. The oxaloacetate that starts the process is different than the oxaloacetate that started the last process. Okay? So the cycle is going to have some interesting effects on the oxaloacetate. But so we've got a 4 carbon molecule again, still very energy rich, and a kinase is going to come along, do substrate level phosphorylation, and convert this succinyl-CoA into succinate. So substrate level phosphorylation. You've got a GDP that bumps into an inorganic phosphate. The kinase pushes them together, and it does that in the process of this energetically favorable reaction of going from succinyl-CoA to succinate. So the delta G here is going to be negative. It's going to be favorable to go from this to this. It's going to be favorable enough that nature has been able to hook up this reaction to a substrate level phosphorylation. Now, a pause. What's up with this GTP stuff? Right? Because we've always been talking about ATP as opposed to GTP.
Is that guanine triphosphate?
It is. So the G is short for guanine. A is short for adenine. You got your C for cytosine, and your, which one am I missing? G-A-T-C. Which one? G-A-T. Oh, T. I haven't said anything about T. Threonine. It's a thymine. Guanine, adenine, thymine, cytosine. All right. So GTP. It's the same nitrogenous base that gets used in making DNA, right? Just as ATP is the adenine that gets used in making DNA. Energetically, GTP and ATP are exactly the same, right? They're both triphosphates. The energy is being stored in smushing together those phosphates at the end of the triphosphate. So GTP and ATP are energetically equivalent. It happens that it is GTP that gets used, or GDP that gets used to capture that phosphate by this particular kinase. But because they're energetically equivalent, it's possible to use GTP to make ATP. So if you prefer, you can argue that what's going on here is that we have a substrate level phosphorylation that results in a net gain of 1 ATP, right? What's really happening is you have a substrate level phosphorylation that creates 1 GTP, which can be used to make an ATP, all right? So a little bit of bookkeeping stuff going on there. Then, one more dehydrogenase is going to be doing its thing to convert succinate to fumarate. Right? We take a high energy electron off and tack it on to FAD. Let's take a look at this structure. See what you think. Is fumarate really that much less of a high energy molecule than succinate is? Well, The energetics suggest that it is, right? This is a circle, it's a cycle. You go from succinate to fumarate. You've stripped off a high energy electron and brought it along and made this FADH2 with it. What do you think about this structure? We've got, what, 1, 2, 3, 4, 5, 6, 7 high energy bonds here. What do we got over here? 1, 2, 3, 4, 5, 6. Hey, how about that, right? There's one fewer high energy bond with the fumarate than there was with the succinate. You guys see that? Maybe a little bit hard to see. I'm squinting, I'm looking real close here. The arrow right now is pointing to what's a double bond. So there's 2 bonds between this carbon and this carbon. But there's a total of 6 high energy bonds here, only 7 high energy bonds here. If I were to show you this molecule next to this molecule side by side and ask you which one has more energy, I think you got to go with the one on the right, the succinate, as opposed to the fumarate, because fumarate has 6 high energy bonds, whereas succinate has 7. Right? But it's also consistent with this idea that we've taken an energy poor molecule, FAD, and we've made it energy rich. We've stripped off some high energy electrons and we've tacked them on to the FAD. All right. Next, 7th. There's another water molecule that gets added on to make this malate. Not a lot of energy transfers going on here. And then one last dehydrogenase does its thing, and we end up coming back to oxaloacetate. Check out the number of high energy bonds in oxaloacetate. I'm having fun with this. 1, 2, 3, 4, 5. Right? How about malate? 1, 2, 3, 4, 5, 6. Isn't it cool the way you always go from more high energy bonds to less? That's what thermodynamics is all about. So you've gone through a cycle. You've made a fresh oxaloacetate that's different than the oxaloacetate that was present at the start of the cycle. Right? So the layout of the carbons has changed, but the bottom line is, check it out. We've released 2 CO2s. We started with a 2-carbon molecule. We get rid of 2 1-carbon molecules, so the bookkeeping works there. And most importantly, from a cell's perspective, is we've gotten some energy in a useful way at several steps in the process. There are 1, 2, 3 NADHs that got made, 1 FADH2, and 1 GTP. The GTP is pretty obvious. There's a high energy bond there. We can do stuff with GTP like make ATP. What do we do with the NADHs and the FADH2s? Well, now, boy, once again, I forgot that I had zoomed in versions. I made you guys squint at the big picture. Sorry. So, what do we do with these high energy molecules like NADH? You know, the PowerPoint slides are available to you on Pilot, so you don't have to squint if you look back at this later on with the actual pictures. What do we do with this high energy molecule? I'll tell you what we do with that high energy molecule. We run it in FADH2 through the electron transport chain. Oh my gosh, guys, there's could be an earthquake.
This is a test of the right state emergency notification system. This is only a test.
So at this point,
this is a test. This is a test of the right state emergency notification system.
What?
This is only a test.
it's just a test. If this was a real earthquake situation,
This is a test.
they're still testing
This is a
this.
test of the right state emergency notification system. This is only a test.
Yeah, it's a 3-minute test, by the way. Now, if this was an actual earthquake, what we would be doing is we'd get low, we'd go under the podium or a desk or go to some of the tornado structures. Again, don't panic and run around in circles, right? Probably not a good idea. But so that's what would happen. While we've got this little test going on, let me tell you something that I think is kind of funny. My 5th grade daughter last night was asking me, she's telling me, he's like, Dad, you know, I've got so many tests in 5th grade. I'm taking like 5, 6 tests a week. how many tests do you take now that you're an adult? And it's like, gee, you know, I don't take many tests at all anymore. Well, today I can tell her I'm taking 3, right? We had 3 tests just now. And then she asked me, what's the most number of tests you ever took in a week? I don't know. I'm thinking maybe 10, right? It could have happened. There was probably a time when I took 10 tests. It's like, wow, well, as a 5th grader, I'm taking 5 or 6 every week. So I'll let her know when I get home tonight that I too took a test today. I was involved with a test of Wright State's earthquake preparedness. All right, well, so, you know, we still got another minute or so of this earthquake test to go through, but I'll take advantage of this quiet time when the earthquake doesn't seem too bad to talk with you about what we do with these high energy electrons in NADH and FADH2. Let's talk about the electron transport chain. There is the electron transport chain in all of its glory. It's a bucket brigade. Plain and simple, that's what's happening here. Check it out. We've got high energy electrons that got captured as part of both glycolysis and the citric acid cycle, residing on NADH or FADH2, but see that high energy electron? It can be handed off from NADH to flavin mononucleotide, amongst friends, FMN.
All clear.
Woohoo!
All clear. The emergency on campus has passed. As information becomes available, it will be posted on the Wright State website.
Don't look for information on the Wright State website.
All clear. All clear. The emergency on campus has passed. As information becomes available, it will be posted on the Wright State website.
You think they're going to do this a 3rd time?
All clear. All clear. The emergency on campus has passed. As information becomes available, it will be posted on the Wright State website.
It wasn't a real emergency, remember, it was a test, so there won't be any information on the Wright State website. But anyway, back to the topic at hand. Now that the earthquake is past, check this out. This is actually a pretty cool bucket brigade as far as bucket brigades go. Check it out. We've got high energy electrons that get handed off to FMN. Okay? FMN gladly accepts that high energy electron and then hands it off to another coenzyme, a cytochrome, in the electron transport chain. Right? Notice what they're doing here. They're trying to give you a sense of the free energy that gives free energy for that electron as it passes through the chain. It starts out juiced up pretty high, right? That one electron with this hydrogen comes in with what, 50 kcals per mole relative to oxygen's electrons, right? So that's a pretty souped up electron. And what happens is it gets passed off, and as it gets passed off, this is an energetically favorable type of process. The delta G will be negative because you go from souped up high energy electrons to a little bit less souped up high energy electrons. And then it gets passed down another step in the process still. It gets passed down another step in the process, and you get this chain of cytochromes, each of which is accepting a high energy electron and handing it off very directly to another cytochrome, but with a little bit less energy in the process. NADH has the highest energy electrons of them all, but FADH2s are no slouches. They come into the electron transport chain in a slightly different spot, so they cut out this part and they come into play here, and they all get passed down, and ultimately that high energy electron gets delivered to oxygen to make the lowest of low energy electrons that you'll see, and that is the electrons associated with water. Okay? So that's what's going on here. Part of what makes this pretty cool is that that electron transport chain is embedded in one of the mitochondria's membranes. So all of this stuff is happening with membrane-bound proteins that are holding on to those cytochromes that are part of this bucket brigade. So it's all happening inside of the membrane. And where the energy harvesting comes into play, if you haven't seen this before, I think this might blow your mind. This is pretty cool how this electron transport chain translates directly into the synthesis of ATP. This is pretty cool. Check this out. It's going to happen because these cytochromes set up something that allows this process of chemiosmosis to occur. And a key player in chemiosmosis is an enzyme called ATP synthase. And ATP synthase is an engineering marvel. Okay? What ATP synthase does, it's a very complicated chemical structure, a protein that all living things have, at least all living things that can do oxidative phosphorylation or aerobic respiration, equivalent terms. But what happens within us as eukaryotes, where we can take full advantage of it, is ATP synthase allows protons to travel from high concentration to a region of lower concentration. And that concentration gradient of the protons is set up by the electron transport chain. The cytochromes and the proteins associated with them, as they're moving through the electron transport chain, kick out electrons from the mitochondrial matrix and push them to a place where they're at high concentration, the intramembrane space. All right, there's a whole bunch of things that are happening in this picture that get me excited at a molecular kind of level of appreciation of what's going on. I can't emphasize enough just how impressed you should be with the structure of ATP synthase. It looks like a turbine in a steam engine kind of thing. It's like, wow, how did you design something like that through random die rolls over the course of a couple billion years? This is an amazing piece of engineering. It looks, for all the world, when you look with an electron microscope at this structure, like the turbine in a steam engine. But there's other cool stuff going on here as well that I think you guys can really appreciate given what we've talked about in this class. Remember, nature doesn't like order, right? The second law of thermodynamics says that you should be increasing disorder. Nature's going to not like, it's going to be pretty disturbed by the fact that there's more protons on one side of a membrane than on the other side of the membrane. Nature wants everything equal. It's a very socialistic sort of way, right, of looking at life. Nature doesn't want that separation that the membrane is causing. It wants the same concentration of stuff on one side of the membrane as on the other. Anything else is too much order for nature's liking, right? So that's freaking nature out. But that's not all that's freaking nature out, because nature also hates separation of charge, right? Those electrons are hanging out on, there's hydrogen ions that are involved with all of this as well. You've got a separation of charge in addition to a difference in the concentration. So it's a double stick in the eye of nature, right? Where not only are we talking here about different concentrations on the different sides of this membrane, but we're talking about different kinds of charges on one side of the membrane relative to the other side. So it's a double offense to nature. And the deal then that living things have worked out that use this electron transport chain and are set up to do chemiosmosis is this. It starts up, it's a pretty clever, involved strategy. It starts out this way. You know, nature, for whatever reason, has been cajoled into allowing high-energy electrons to be tacked on to NADH, and then also FADH too. But let's just focus on NADH. It makes it easier. So nature got tricked, you might say, into allowing NADH to be made, right? And now you got this NADH, and you dangle that in front of nature, and you say, hey, you know, look at the high-energy electron on this NADH. Boy, that's a high-energy electron. I bet you would like it to be a lower energy electron, wouldn't you? Nature says, yeah, I don't like high energy electrons. Says, oh, well, here's what I'm going to help you, nature. I'm going to let that high energy electron become a lower energy electron. Let's hand it off onto flavin mononucleotide, the first of the cytochromes in the electron transport chain. Well, you just hand it off, right? Lower energy, you should like that. And hand it off again, and hand it off again. And, oh, you know what? The next time that I hand it off, what I want to do, if you don't mind, is just take one hydrogen ion from one side of a membrane and put it to the other side of the membrane. Are you good with that? Yeah, okay. All right, you twisted my arm. Okay, good. And we'll keep handing off this electron 3 times as the electron passes through the electron transport chain. Protons get moved from one side of the membrane to the other side of the membrane against a concentration gradient, setting this system up where the intermembrane space has this surplus of hydrogen ions, especially relative to what you see in the mitochondrial matrix. And at the end of all of that, nature is told, see, I told you I'd give you a low energy electron. Look at that. You can't get lower than water. That's as low an energy as an electron's ever going to get. Aren't you happy, nature? And nature says, yeah, I'm happy, but man, what's up with this weird separation I've got somehow or other? There are all these hydrogen ions up here. There are no hydrogen ions down there. And then living things come back and say, oh, that bothers you? Oh, man, how did that happen? Boy, that's a shame. I'm going to help you out. I'm going to help you out by having a protein that's sole purpose is to let those hydrogen ions flow back across that membrane, go from high concentration to low concentration, just because I like you, nature, and I want to help you out. Right? Oh, and by the way, while I'm helping you out, would you do me a solid as well? Because I kind of like these ATP things. I need those for some other stuff I want to do. Would you mind if while I'm letting the protons come across that membrane, if just once in a while, you know, every time, let's say, that the proton comes across, you let me make an ATP? You good with that? Well, nature says, I'd really rather not. Oh, yeah, I know, but man, it's a bummer. You got that hydrogen ion separation going on here. It's like, yeah, you're right. Okay, fine. We'll do the ATP. Great negotiating that has played out over the course of 1 billion or so years of this system being at work. But that's what happens for every hydrogen that's allowed to pass through this ATP synthase across this membrane. A ratchet smushes together an ADP with a phosphorus, an inorganic phosphate, to make ATP, which is payola for the cell. I got my dollar bill of the energy currency made, and all I had to do was allow this proton gradient to drive my pump, to drive that turbine. So, check it out. Here it is, the whole picture now together. You know, we had one slide that showed you just the electron transport chain and some finer detail, and we had another picture that showed you the ATP synthase. Now you can see the electron transport chain is part of the same phospholipid bilayer membrane as the ATP synthase is hanging out. All right? Check it out. NADH, for every one of those high energy electrons that NADH has, you are able to persuade 3 protons to move across the inner mitochondrial membrane. How about that? Every one of those NADHs lets you move 3 protons across. FADH2, a little bit less spectacular. You only get 2 protons moved across as opposed to 3. But for every proton that gets moved across that inner mitochondrial membrane, for every one, when it comes back across the membrane, when this gate that's called ATP synthase allows it to pass through, you turn the crank and you make 1 ATP. Pretty cool, huh? That's where you get the big harvest of ATP. This is called oxidative phosphorylation. It's oxidative. Well, the phosphorylation is because ATP is being made from ADP and inorganic phosphate. ADP is being phosphorylated, okay? But it's oxidative phosphorylation because you really can only do this if you can dump off those high energy electrons onto oxygen to make water. Okay? It's oxidative because ultimately oxygen is going to accept that high energy electron to make water. If you don't have oxygen around to act as that final electron's receptor, this can't work. Okay? And so that's why in aerobic circumstances, when oxygen is present, we can get a whole lot more ATPs from every glucose than we can in anaerobic respiration. That's why things like botulism that can live anaerobically, bacteria that can live anaerobically, just ekes by on 2 ATPs for every glucose that it consumes, right? It's something. I guess, you know, if that's all you got, you're happy with it and you make do, and things that can't do anaerobic respiration can't make do at all, and so botulism can thrive in that circumstance. But if oxygen's available, wow, how much more energy can you get? You can do the math, and you can figure out how much more energy you can get. I'm going to zoom back and see if I can find a picture. Oh, man, how far back do I have to go? Too far back, I think. I want to draw your attention to just how many of these NADHs are being made. The bottom line is this. In the absence of oxygen, glycolysis gives us a net gain of 2 ATPs. In the presence of oxygen, what you can do is you can run that citric acid cycle, you can make a whole bunch of NADH and FADH2, and you get a net gain of 32 to 34 ATPs per glucose. That's the difference that oxidative phosphorylation allows. A total of something like 30 additional ATPs can be made as a result of what's playing out here with chemiosmosis in the electron transport chain. There's the picture I was looking for. It was next in line. All right? So check out what's going on here. Glycolysis, substrate level phosphorylation lets you make 2 ATPs. If that's all you got, it's better than nothing, right? The citric acid cycle, remember, there is that one substrate level phosphorylation. The citric acid cycle is going to crank 2 times for every glucose molecule. So you'll make 2 GTPs, which can be converted to ATP. Not too bad. 4 at substrate level phosphorylation. If that's all you got, I guess you're happy with it and you make do. But whoa, if you can use your NADHs and FADH2s to drive the electron transport chain, now we're talking about the big payoff here of either 26 to 28 more ATPs to give you a net gain of 30 to 32 ATPs. They say about, and the numbers are a little bit fuzzy because some days it works better than others, some systems are set up better than others, right? Particulars aren't that important. It's just the relative scale of things. You get like 10 times as many ATPs made from chemiosmosis and oxidative phosphorylation as you do from substrate level phosphorylation. Okay? Little bit more about what do you do if you can't do oxidative phosphorylation, right? Because for many organisms, and even for you and I, in certain circumstances, our cells find themselves with not enough oxygen to do the whole kielbasa and collect all of that ATP that you would get from chemiosmosis. All right? So what do you do in the absence of oxygen? If you're a bacteria, what you would do is you would run through glycolysis, you'd make your pyruvate, and then if no oxygen is present, you do this thing called fermentation. One last attempt to try to get some ATPs out of pyruvate. And what will happen with fermentation, you convert this 3-carbon molecule pyruvate to a 1-carbon molecule called carbon dioxide and a 2-carbon molecule that is many people's favorite molecule of all, ethanol. Right? To lay people, alcohol, booze, hooch, right? That's what's going on with fermentation. If you wanted to make beer, or you want to make wine, or any other spirit, right? A big part of the process is you gotta get some sort of microorganism, yeast much more often than not. You gotta persuade yeast to go through glycolysis. Here, you want some sugar, you little yeast? Have fun. I got a whole bunch of sugar for you, but I'm not going to let you use any oxygen to extract all the energy that you might have been able to extract. And so what does the yeast do in the absence of oxygen? It drives it through, it takes the sugar through glycolysis, creates pyruvate, and then says, okay, now I'm ready to go. Let's use the citric acid cycle. But wait, I can't because I don't have any oxygen. If I don't have any oxygen, what can I do? This pyruvate's juicy. It's got some energy in it. Let's take off one CO2 and use that process to make a couple ATPs and then alcohol as a waste product. Okay? Alcohol's relatively energy rich, but it's a waste product for the yeast because it can't take it the rest of the way through the citric acid cycle, and you got a couple more ATPs, maybe a total of 4 ATPs per glucose, and you've made ethanol. You and I don't get drunk when we exercise anaerobically. Times that I feel like this is a disadvantage that we have. Instead of doing like yeast and making ethanol as the end of the line, we make a 3-carbon molecule called lactic acid, or the salt of lactic acid is lactate. In lactic acid, we get another 2 ATPs out of the process, so we go from glucose, substrate level phosphorylation through glycolysis gives us 2, converting pyruvate to lactate gives us another 2, so we got 4 ATPs. If you need energy for the cell to drive the actin and myosin in your muscles to make things move and to do all, if you need it, but you need it so fast that you don't have the time to deliver enough oxygen to run things through the citric acid cycle, you'll take those 2 extra ATPs, thank you very much, and you make lactic acid. And physiologists are debate about this, but I know there's a popular school of thought that says that the reason your muscles are sore after anaerobic respiration, after weightlifting, is because the lactic acid forms little crystals in your muscle cells that poke your nerve cells and make you feel like you don't want to do that anymore. And again, you know, ethanol probably would have worked better for us in some sense, but for whatever reason, we're making lactic acid as a result of anaerobic respiration. Right? In much the same way that yeast and other bacteria would undergo fermentation to make ethanol. But when you get oxygen present, don't be making lactic acid. Bad idea. Nobody wants to have lactic acid in their muscle cells. Instead, run that pyruvate through the citric acid cycle, and then ultimately crank through the oxidative phosphorylation that you can do with chemiosmosis and really get a lot of ATP that way. So there's a fork in the road. If there's no oxygen present, you got no choice but to go down this path. If there is oxygen present, you could go either way. It would just be a bad idea to take this path as opposed to this path, because you get like 10 times as much ATP when you take this path as you do when you take that path. Now, what is this slide about? Oh, well, this is interesting, right? It's showing you that when you're making ethanol, what you can do is this is a nice, easy way to regenerate the NADH, or the NAD that you need to drive glycolysis. There is a step, there are some dehydrogenases that work. There is a dehydrogenase that works in glycolysis, and so that's why fermentation, when you're making ethanol, what you're doing is you're really just trying to regenerate those NADs that you're going to need. And the same is true then for us when we're making lactate. So we've already talked about this slide. I don't think I need to say anything more about it, but was it a week and a half ago, I talked with you guys about enzymes and some of the wondrous things that enzymes can do. I wanted to toss this at you just to point out that, again, enzymes catalyze these pathways, glycolysis, the citric acid cycle, and enzymes are wondrous things. They speed up the rate of reactions by lowering activation energies, right? They're also wondrous in that cells can control the activity of these proteins, okay? And so what you see here is that, you know, it maybe would let you appreciate for the first time that there may be times you don't want to do glycolysis. If you've already got a lot of ATP, keep your powder dry and leave your glucose alone, right? Every time you go to the bank with your glucose, you're getting screwed in the exchange rate. All right, so if you already got enough ATP, I don't want to mess with it. Hang on to it. Keep it just the way it is. Thank you very much. All right, because what happens when you get a lot of ATP? If you got a lot of ATP, cells start to make glucose out of it. You're like, oh, that's a bad deal, right? You know, so you're better off just keeping your glucose the way it is if you already have a lot of ATP. So check it out. AMP is adenosine monophosphate. You know what a cell does when it's really starved for energy? It converts its ATP to ADP, adenosine diphosphate. And if it's really hurting for energy, it takes energy out of ADP and makes AMP, adenosine monophosphate. If AMP levels in a cell are high, that's a pretty clear sign that this cell is hurting for energy, and it might be a good time to crank up glycolysis. Get phosphofructokinase, the very first enzyme in this metabolic pathway, juiced up, doing its thing more and better to start driving things through the pipeline. But by the same token, if you have a lot of citric acid, that should be a message that says, hey, feedback inhibition, guys, let's do that thing. We already got enough citric acid, more perhaps than we can work with. Don't be making any more. Where do you do, what's the best place to stop this? Before the glucose gets out of the barn door, shut it down before you even begin the rest of these processes. And by the same token, ATP is an inhibitor for phosphofructokinase. Again, sending the signal that says, hey, we got a lot of ATP, we have a lot of energy, we don't need to convert more glucose into ATP, right? If you were designing this deliberately as an engineer, sitting down, paper and pencil, figuring out how to best game this system, given the players you've got, that pathway of glycolysis, and what's involved with the second law and the first law of thermodynamics, would you have designed it any differently, right? Pretty nice bit of engineering here, don't you think? Because the place to do it is at the very start, right? You could have, if you wanted to, inhibited any of these enzymatic steps. Why would you do that, right? Why would you stop it here as opposed to stopping it right at the very beginning? And you could have turned on at any of these steps. Why would you do that? Here's where it really is important when you start things off in the pipeline. Getting things into the pipeline at the beginning or cutting off the pipeline at the beginning, that's the way to do it. And what molecules would you use for giving that sort of feedback? Well, ATP is a no-brainer. Citrate, relatively no-brainer. may not have contemplated this one. You might have thought ADP as opposed to AMP, but AMP is just ADP with an exclamation point next to it, to the cells. And so if AMP is an option, that's a no-brainer as well. All right, so again, what a neatly designed system we're talking about here. All right, well, we're at the end. On Tuesday, I'm going to talk with you all about the flip side of this. We're going to talk about photosynthesis, how you make those glucose molecules in the first place. But that's what I wanted to talk with you guys about with respect to catabolism and glycolysis in the citric acid cycle. Here's a question that we'll have on your next test. And have a good weekend. See you guys on Tuesday.